All right, well, welcome to High Tide Aquatics for another cocktail and coral talk event with Mr. Charles Delbeek. A little bit about Charles, if you guys don't know who he is the author, co-author of probably 3 of the most important books in this hobby, the Reef Aquarium series that he co-authored with Mr. Julian Sprung. He is currently the Curator of Aquarium Projects at the Steinhart Aquarium at the California Academy of Science, where he helped design and consult on their 212,000-gallon Philippines reef system, which he has been running for 16 years now. Prior to that, he spent 13 years at the Waikiki Aquarium in Honolulu. He is a member of the Zoo and Aquarium Association. He was also a 2008 recipient of the MASNA Award. So we are pleased to have Charles here. I want to thank everybody for registering and donating to help keep this awesome event going. I want to thank my sponsor, Bay Area Reefers, local reef club. I've been a member 17 years. Local reef clubs are a great thing for this hobby and industry, so keep supporting them, keep them going. With no further ado, let me introduce Mr. Charles Delbeek.
Oh. Thank you, Ken, and I'm very happy to be here when it's cooler. Last time I was here, it was quite warm, and we had somebody pass out, so I was thinking maybe it was because my talk was too much. I don't know, but it was the temperature in the room. So today I'm going to talk about, or tonight, I'm going to talk about something that I've been looking at for probably a couple of years now, and why I think we were having a lot of trouble with our tank. And it was something that I kind of stumbled upon. I had a kind of a suspicion that was an issue. And then I started looking into the scientific literature and reading up about it more and speaking with some scientists. This past summer I was in Monaco, and I went to the Centre de Scientifique in Monaco, which is one of the research centers on coral nutrition and the effects of nutrients on corals. So I met with Dr. Christine Paget-Verrier, who is an author of a lot of the papers that I was reading, so it was really cool. Well, of course, I went to the aquarium as well, and because that's where I was inspired by Jacques Cousteau when I was a kid, watching the documentaries in the 60s on TV. I saw the beautiful colors on the Red Sea coral reefs, and that's what I want to do. So I knew from the age of like 9 or 10 what I wanted to do with my life. And a couple of detours along the way, but I kind of ended up where I wanted to be. So we're going to talk about nutrients, and mainly nitrogen and phosphorus. So first, a little bit of background on our tank, for those of you that are not familiar with it. So it was first built, started in 2004. We opened in 2008. So it's been, as Kenny said, it's been running for 16 years. It's 212,000 gallons. The deepest point is about 25 feet. It has 5 windows. Big main window that you can see here in the photo. And we also have a shallows area that you can look at from the railing, which is above that seating area. The first fish and corals were added back in March of 2008. And currently, in terms of lighting, we have 72,000 watt metal halides. They are the ones that you see here on the catwalk they're all on electronic ballasts now, but these ones on the catwalk, not all of them are running at 1,000. There's probably like 6 or 8 that run at 1,000. The rest run at 750. And now we've also been working on adding Kessel lighting. So we're using the W2K lamps. We have 6 of those in operation. And I also have 4 eco-exotic 460 nanometer LEDs that are on all day long, and also for evening events. And there's a mixture of 10,000 and 14,000 Kelvin. Filtration, we have 4 25 horsepower pumps that you can see there on those concrete stands. 2 of them supply water to our 4 sand filters, and the other 2 supply water to our 2 large MRC fractionators. And the turnover of the tank is about once every 45 minutes. So it's a little bit higher now, it's closer to 60 minutes. We also have 3 40 horsepower pumps. Here's our fractionators. And these are the 3 40 horsepower pumps that are on a closed loop basically. And they send water out to 5 different areas in the tank. I also have 5 hydro wizards in there. We also have a media reactor system here. We have 2 large 8-foot calcium reactors. And we have some media reactors and GFO reactors. We also use ozone, and we use lanthanum chloride. and we are also adding some trace elements. So the ones that are in black are ones that we add to our salt mix. We make our own salt water. The ones in blue are in our tap water. So the manganese in the tap water is about 2 parts per billion. It's quite high compared to what we would normally want in a tank. The iron is pretty high too, but by the time you mix it with the salt, it all precipitates out because it's just free iron. The carbon, here's those 3 pumps. And I've also been working around with the potassium iodide, manganese, and iron we add. We've been adding for a long time, but I'm also starting to add molybdenum, cobalt, zinc, barium, vanadium, bromine, and fluoride as well. Trying to get those levels up, and we'll see what happens. Hopefully I won't kill everything. We haven't used carbon since July of 2013 when the carbon reactor broke. We rebuilt it, but we never turned it back on because we didn't see a need for it. And we have not used GFO since November of 2023. It's a pain in the ass, especially when we have these large reactors. We're using like 150 or 200 gallons worth of this stuff. It's a real mess to deal with it, so screw it. I can do it with lanthanum, works just as well. So as you all know, as being reef hobbyists, when you operate in a tank like this, it's like a balancing act, right? You're always trying to balance things. You're balancing your water chemistry, your lighting, your life support system, or in other words, your filtration. You have pests and diseases that pop up that you have to deal with. And then, of course, you have the unexpected stuff. Things you're not anticipating, that happens as well. So you have to deal with that. Pump breaks, this breaks, that breaks. You get an outbreak of RTN, you get an outbreak of brown jelly, whatever. And so it's no different in a tank this size. It's just that it happens on a much bigger scale, and it takes a long time to fix it. And we kind of liken operating a tank like this like steering an oil tanker. Little tiny changes can lead to big changes down the road, right? So a little change in course can put you 10 miles off your destination if you're doing an oil tanker. Same with these big tanks. You make a little change, you're not going to see anything for a while, and then you're trying to turn it around, it's going to take you just as long to turn it around again. So one thing that happened to us just before COVID started, we had a massive outbreak of brown jelly. And we used to have a whole wall full of echinopora, and the brown jelly basically came in and wiped out everything. So we had to be pulling it all out, and then COVID hit, and so we couldn't really deal with that and how to get it back again. So that was a big problem. So that's the first clue that something wasn't right in the tank. Second clue was, by August, we started to see a lot of corals that had started to bleach. They were losing color. So this is a Diploastria, and you can see it's lost a lot of its color. Excuse me. However, within 7 months, it was starting to regain that color again, and it's starting to grow again. And by April, a lot of the corals are looking really good. So these are some of the colonies that we had in the tank at that time. The other thing that we've had the opportunity to do, which you guys can do pretty easily, just turn the light, just go down in the living room in the basement, look at your tank at night. We have to dive in the tank at night to see it, so we do night dives in there. So these are the kinds of things that we see at night that we didn't normally see during the day. So you have Tubostrea that had settled out in the rocks that was growing out. In the middle you can see sweeper tentacles on echinopora. They're quite large, they're about that long. So all these huge plates that we had were putting out sweeper tentacles, probably twice that area of size. And just a lot of the other corals that were opening up to feed at night. We also had a lot of times issues with Montipora foliosa. A lot of people call it Monty Cap, but it's not. It's actually foliosa. And within a couple of years, that took longer to come back, but eventually did come back. So we thought, oh, everything's great. So I go on vacation, and just before I go on vacation, my biologists are telling me things aren't looking good. And so these are some examples of the things that they were seeing, and we're going to concentrate on this guy in the corner we called Sad Sack. Sad Sack was not looking good. So when I came back, I said, okay, let me start digging into what's going on. So the first thing I did was I looked at my nitrates and my phosphates. So back in COVID, when we had this issue with the corals lightening, my first instinct was to say we don't have enough phosphate. So we stopped adding lanthanum to allow our phosphate to go up. The other problem was that we, because it was COVID, we didn't have dive shows going on. So we didn't have those feeds going in. A lot of it's pellet food, which is high in phosphate. So that phosphate wasn't going in. So our phosphates were getting, I thought, were getting too low. So here's a graph. The nitrate is the orange line, the phosphate's the blue line. And you can see in August that our phosphates started going down, all the way down to about February, March, April, that whole area. Why were they going down? Well, we found out that our lab had made an error in their calculations, and our phosphates were actually 10 times higher than we thought they were. So instead of 0.07, it was 0.7. So we thought, oh my God, that's way too high, we better get it down. So we upped our lanthanum dosing, and we've brought the phosphate down. And you can see at the same time, the nitrates started to accumulate as well, because we were feeding the fish more. So that's the first clue. Something weird's going on. So we got that drop in phosphate, and we had this increase in nitrate. And we get this inflection point around January. And then by May, June, July, we had this big gap between the two of them. And this is important. So we'll come back to why that's important. So then I started thinking, well, what happened back in, just before COVID started, when we had the issue with the brown jelly? So I looked at those phosphate numbers, and I saw the same pattern. I saw the phosphate going down, the nitrate going up, and we got this big gap right around September or so. So things were going south already during COVID. And then they started to narrow up again because we let the phosphate go up. So I remember I said, hey, let's stop adding lanthanum, let's let the phosphate go up. So there's a lot of talk about nutrient ratios. And the one that's usually brought up is this Redfield ratio, right? Everyone knows the Redfield ratio or have heard of it. It's basically based on work done in 1934 by an oceanographer named Redfield, who worked with oceanic phytoplankton. And what he found was that they used up the three main nutrients for plant growth, carbon, nitrogen, and phosphorus, in this ratio, 106 to 16 to 1. Most people focus on the 16 to 1 for nitrogen and phosphorus, right? So the problem is, first of all, this is an atomic ratio. It's a molar ratio. It's not based on mass. So you can't just take your nitrates and your phosphates and divide them and get this ratio. That's not how it works. The other thing is that this is a ratio for phytoplankton. It's not the ratio for coral. So coral contains animal tissue and it has unicellular algae, dinoflagellates. So they're a little bit different. And there's the ratio that's in your water, and there's the ratio that's in the tissue of the coral. They're different. So what you can do is you can take your nitrate and your phosphate and you can divide them to get a ratio, and then you multiply that by 1.353 and it gives you your molar ratio. So let's say your nitrate was 15 and your phosphate was 0.45, divide it, multiply by 1.53, and you get a molar ratio of, yeah. I think I'm missing something here. Let's see here. Oh, shit. Okay, I'm missing one very important slide, which basically was going to tell you that the ratio for corals is about 50 to 1. For cyanobacteria, it's 5 to 1. For bacteria, it's 10 to 1. So all these things have different ratios. So that's why when you add alcohol or some kind of carbon source, it's taking it out, the nitrate to the phosphorus at a 10 to 1 ratio, because that's what bacteria use it up at. So I was using a molar ratio because that's what all the scientific literature talks about, for the most part. It's all in moles. So I can calculate my molar ratio by doing this. And so that's a good ratio, I would think, because it's very close to the ratio that corals have in their tissues. But you guys don't deal in those, right? So you deal in milligrams per liter. So some of the testing labs, like Fauna Marine, will give you a number. So if you do an ICP test with Fauna Marine, they will give you a range of 90 to 110 for your ratio. And so if you had a nitrate ratio of 40 and a phosphorus ratio of 0.4, your ratio would be 100, which would fall within their range. The problem with this, that's good, but this is not a nitrogen to phosphorus ratio. This is a nitrate to phosphate ratio. So it's not the same. It's close, but it's not the same. And nowadays, a lot of people are also adding ammonia to their tanks as a nitrogen source. So that's also going to change this around. And so this is the kind of thing that you get from fauna marine. And basically here it shows you this ratio. Does anybody see what's wrong with that? That is phosphate divided by nitrate. That is not nitrate divided by phosphate. So they know about it. I've talked to Claude. I pointed it out to them. They're trying to fix it in their software. The number's right. The calculation's right. It's just that that's backwards, which makes me wonder about that one and that one too. But I don't know. I haven't looked at those. So just buyer beware, right? When you get these numbers, look at them with a bit of a critical eye. So this is actually our reef tank. So our nitrates were around 41.7, and our phosphates, if you measure it with a test kit, this is what you would get. This is actually the total phosphate because they measure, when they do ICP for phosphorus, they're measuring inorganic and organic phosphorus. So that's everything together. And you can see, this is where my measurement values are in red. They don't fit on their tables. They're like way off. This is where they're supposed to be, according to them. But my ratios are my numbers are nowhere close to that. So these are the tissue molar ratios for corals, which is an animal plus bacteria that live in the microbiome, the holobiont as they call it, the zooxanthellae, it's about 50 to 1. Green algae, dinoflagellates, 20 to 1. Well, you can read the rest for yourself. So the Redfield ratio only applies to phytoplankton. So what I did then is I took my nitrate and phosphate numbers and I calculated this molar ratio. And you can see that it started to climb in about January. So by June, when we took that photo, those ratios were pretty high. Not looking happy. But we started to bring it down because we let the phosphate go up, and the corals started to heal up again. So then I look back at the same kind of graph. This is my nitrogen phosphorus ratios for that period during COVID. And here's when that coral was not looking good. And here's when the coral recovered, and also the other corals look good. So there you can see that my ratio is close to 50 to 1. So then I thought, okay, I remember we had a huge die-off between 2012 and 2014. We had 881 corals in that tank at that time. Within 2 years, we had lost over 300 of them. So I thought, okay, let me look at those graphs. And what do I find? The same kind of trend. Phosphate going down, nitrate going up, getting this huge gap in between. So basically, the phosphate is the denominator, right? So as that denominator gets bigger, your product, your division answer is going to get larger as well. And so then, okay, let me graph my molar ratios. And again, I see these huge spikes right here. So I thought, okay, I did all this stuff. What about other people? What are their tanks? What are their numbers like? So this is Georgia Aquarium's coral tank. This is what it looked like last summer, 2023. And I did have their data from when they first opened in 2005 up until 2009. So these are their nitrate values and their phosphate values. This is the phosphate, sorry, it didn't turn blue. But you can see that their phosphate values kind of were between 0.4 and 0.1 in that area. But their nitrates went way up. Their nitrate values were over 100, where the phosphate just stayed low because they were using phosphate removers. The other thing that when I visited them in, I think it was 2012, I met with the curator of that tank and she was showing me their nitrate numbers. And all it said on the head of the readout that she get is nitrate. Not NO3 or anything, just the word nitrate. And I asked her, well, what is that? Is that nitrate or is that nitrogen? So basically nitrate is the nitrogen plus the oxygen. Nitrogen is just the nitrogen portion. It's still the same amount of nitrogen, but it gives you a different number. So she went back to the lab, and it turns out that the number they were getting was nitrogen. So instead of having a nitrate value that she thought was around like 15 or 20, it was actually 80 for nitrate. So anyway, you can see how it climbed up like that. And they were struggling to keep a lot of corals. Some corals were doing okay, others not so much. So let's fast forward. So you can see it goes up there, and then they got that big gap as well. So I did their molar ratios, and you can see their molar ratios were as high as 1,200 in some cases. So then Steve Harder's now the person who's in charge of that tank. He's been working on that tank for probably close to 8-10 years now. And so I was able to get numbers from him for the last 4 years. And you can see a huge difference here in terms of the nitrate. The nitrates are pretty low. They're below 10 or 12. The phosphates are still around the same range. And then you'll notice here that they're both going up. So I asked him, well, why was your nitrate so much lower? He said, we got rid of all the big fish. So they had sharks in there. They used to have big groupers and all kinds of stuff. Sweet lips. A lot of the big fish, they went out, so less food was going in. The phosphate, they had not been using lanthanum or anything to control the phosphate because it was staying low, because they had so much coral it was sucking it up. And then they started to add more fish and start to feed more, and you can see that both numbers are going up. But that's the key. Both numbers are going up. Not just nitrate or just a phosphate, both are going up. So when you look at their molar ratios, you can see that at the end, even though both are going up, their ratio is 50. All right, here's another one. This is the Burgers Ocean coral tank in Arnhem, the Netherlands. It's at the Burgers Zoo. This is a 180,000 gallon live coral tank. And it's run on a very different philosophy in terms of very minimal filtration. The whole point is that the tank, the animals, the rockwork, the sand, that's your filter. So they have minimal skimming. They don't add a lot of stuff. They're adding some trace elements. They don't use sand filters anymore. They use drum filters. So I got some of their numbers, and you can see how low their nitrates are. And their phosphates are a little bit higher. But again, when they go up, they both go up together. When they go down, they both go down together. And when you do their molar ratios, they're all below 50. Sounds convincing, right? So then, how many people know ReefBum, or the channel ReefBum on YouTube? People watch that. Yeah, Ken Bokrohama. So he posted a video about issues that he was having with his Acropora. So I reached out to him and I got his phosphate and nitrate numbers. So this was 3 types of coral that he had in March that were all paled out. His phosphate was 0.01. And his nitrate, he had 2 systems. One was 5 parts per million, one was 10 parts per million. His ratio on the 5 ppm tank was 765. On the other tank, it was 1,530. So both tanks were unbalanced in terms of nitrogen and phosphorus, and basically the corals are phosphorus starved. They don't have enough phosphorus. So then he started to up it. So by April, things started improving, and by May, everything was looking much, much better. He was very happy. Things were growing, colors were back. So what was going on here? So here you can see from March to April to May for this coral. So over a span of 8 weeks, he increased his phosphate by 10 times. So he increased his phosphate to 0.1, and now his NP ratio was 76.5. The other one was 153. So much, much better. Not 50 to 1, but they don't necessarily have to be 50 to 1. So both tanks are now balanced in terms of nitrogen and phosphorus. They're no longer phosphorus starved. Then I reached out to a friend of mine who is working with these Florida reef trap corals. This is part of the rescuing corals from the Florida Keys and that are being decimated by stony coral tissue loss disease and holding them in systems. They actually were able to buy the entire setup from Worldwide Corals. They now have their old warehouse and keeping corals there. And he was telling me that, you know, there's a bunch of aquariums and other institutions around the country that are housing these corals. And he said other people were running their nitrates at 5 ppm and his was only 0.2. So he thought, well, I want to raise mine too, because everybody's having really success. So originally his nitrate was 0.2, his phosphate was 0.03. So his ratio was 10.2, which is very close to natural coral reefs. He raised it to 5, which put his ratio to 255. And he said, I don't know what happened, but the corals didn't look good, so I went back again to the lower nitrate level. It's because they were being starved for phosphorus, and also they did it so quickly. So what's going on here? Why is this happening? How can you have a phosphate that's high and then say that the corals don't have enough phosphate? So basically, when you have an imbalanced ratio, so if the nitrogen, for example, is much higher than the phosphorus, the nitrogen increases the number of zooxanthellae. It's a nutrient for algae, right? So you'll see the corals will get darker, they'll get more zooxanthellae, or they'll make more chlorophyll within each zooxanthellae cell. And that increases the amount of photosynthesis that's going to happen. Therefore, their demand for phosphorus is going to go up. So as you increase that nitrogen, they want more phosphorus. So when that phosphorus is limited, then the photosynthetic pathway starts to break down. They become less efficient in photosynthesis starting. Also, because they're so hungry for their photosynthesis is kicking in and these cells are growing, where are they going to get the carbon from? Normally the carbon comes from the coral. They're taking it from the interstitial waters inside the coral. What is the source of that carbon? Carbonate. So now they're going to steal that carbonate from the coral. So now the coral has a hard time calcifying. It may still grow, but the skeleton is going to get really brittle and thin. Or they'll stop growing. That's the other option that can happen. We'll get back to how we can get around that. Let's say you had the opposite case. Let's say you lower your nitrogen too much and you got a lot of phosphorus. Again, you're going to get an increase in the number of zooxanthellae. And it might also result in an increase for need for some more nitrogen. But what's interesting is research done on this has shown that the photosynthesis still remains efficient. Corals are adapted to low nitrogen levels in the water. They have ways of getting it. There are bacteria that will fix nitrogen for them. You can get nitrogen from the food and phosphorus from the food. So there's lots of different ways to get it. So low nitrogen levels don't appear to impact the coral health as much as low phosphorus. As one researcher told me, as long as they have some phosphorus, they're happy. Nitrogen is not as much of an issue. So these are some images and some data from a paper. So this is basically what they were calling nutrient limitation. This is what you would find on a normal reef: low nitrogen, low phosphorus levels. So this is Montipora foliosa. So now let's say you add nitrogen and phosphorus together. So you're increasing both, like in the Georgia Aquarium and in our aquarium. What happens? Does it look familiar? The coral gets darker. Why? Because it's making more zooxanthellae. They're multiplying. The coral has lost control. Normally corals can control the number of zooxanthellae by limiting how much nitrogen and phosphorus they deliver to those zooxanthellae. However, when you give them lots of it, they say, screw you, coral, I'm going to get it right from the water. And now the coral's lost control. What about if you just add nitrogen without increasing the phosphorus? Or let's say you take your phosphorus down, like Keith did, got it too low. What happens? Well, now you have phosphorus starvation. And that's what the coral then will look like. So you have high nitrogen, low phosphorus. Still not convinced. Here's another one. So this is Fimbrophilia paradivisa. Kenny's got some for sale here. Here is what it looks like with high nitrogen and high phosphorus. All those little blue circles are zooxanthellae. lots of zooxanthellae, the coral looks fine. But you take away the phosphorus, this is what happened to those fimbrophilia. Zooxanthellae are fewer, but they're larger, but there's less of them. So probably they're making more chlorophyll, they're enlarging in size, they're trying to make up or to be more productive, but they can't make more because they don't have enough phosphorus. All right, well, what happens in the wild? So these are some NP molar ratios that I calculated based on nitrogen and phosphorus data from different reefs around the world. So you can see they're all pretty low, meaning that the corals are nitrogen limited and phosphorus limited. They're under control. Then you go to the Florida Keys. Surprise, surprise, the numbers are going up. Why? Because of the amount of nitrogen that's going in there. From runoff from the Florida Everglades, from all the fertilization that's going on. That's why they've been pushing for decades to basically put the Everglades back to the way they were and before they did all the agriculture to try and combat this. So these numbers have been going up, and there's a paper that documents this over 30 years, how this ratio has climbed over the years. What's interesting is the inshore waters are lower than the offshore waters. And then the same in the Bahamas. You've got some high levels there. So these are most likely because of anthropogenic effects, like man-made effects, us dumping in nutrients. But there are also, in the wild, examples of natural increases in nutrients. And there's a study done in Fiji on 2 islands. And here you can see they have this giant NP ratio. Why is that? Because what normally lives on these little tiny coral atolls and little islands? What kind of animal usually lives there? That nests there? Birds, seabirds. And what do seabirds do? They shit everywhere, right? And that's nitrogen and phosphorus. So this study looked at these 2 different islands. So one of them basically, this is a seabird to seabird and seabird to rat. What do rats do? Eat the birds, right? So this is basically, the upper line is an island where there were no rats, just seabirds. The one below it were seabirds where there was lots of rats. And they took corals from the seabird colony and put it onto the island that had a lot of the rats, so it had less seabirds. And then they left the same coral on the seabird island. So you can see the growth rates between the 2 of them, here and here. Same age, everything. Just that this one has more nitrogen and phosphorus. Same thing here. This is where they took corals from the rat island and put it on the seabird island and left ones on the rat island. So the rat ones, again, didn't grow, just like these. But if they took that coral and put it where the seabirds were, they grew like crazy. So it happens in nature too. So we talked a little bit about carbonates, how when you have these high nitrogen levels and phosphorus levels, they're going to steal the carbonates from the coral. So carbonate alkalinity. So when the nitrate is high, the zooxanthellae are going to use inorganic carbon, and they get it from bicarbonates. So they're going to outcompete the coral for carbon, which limits their ability to calcify. So therefore, the elevated levels of carbonate alkalinity provide the dissolved organic carbon needed for calcification. That's why, and this was back in the late 90s and early 2000s, there was a researcher by the name of Marlon Atkinson at the University of Hawaii, who I knew quite well, who was really puzzled about how can these hobbyists keep all these corals in these, for him, high nutrient waters, even though our phosphate was 0.01 and our nitrate was 5. Compared to the ocean, that's still pretty high. He couldn't understand it. So basically what they found was if you up the alkalinity, now they have enough. So now they can calcify. So that's why it's important, it was important back when we were keeping these higher nutrient levels, that we also had a higher than normal alkalinity level. What happens though, what can happen in some cases is that those corals will continue to grow quickly now. They've got all that carbon, the tissue is growing really fast because of all the nitrogen and phosphorus. The coral has to keep up with that tissue expansion. What happens is the phosphorus also gets into the skeleton and it makes a less dense skeleton. So when I worked at the Waikiki Aquarium, we had well water, which was high in nitrogen and phosphorus compared to the ocean. And we could grow big heads of Euphyllia and Acropora, but you could take a Euphyllia skeleton that we had grown, say, for 5 or 10 years, and you could crush it in your hand. You could take Acropora and pinch it and just turn it into powder. That's how less dense it was in the wild. So Bruce Carlson, who was the director and had collected a lot of these corals in the wild, was a smart guy, and he had actually had GPS coordinates for every coral that he ever collected in Fiji and Guam. So he had Calasia fricata, the candy cane coral. We had been growing it for years at Waikiki. He went back to the exact same colony that he had collected 15 years from before, collected samples, brought them back, and they did a study, and the ones that we grew were 50% of the density. Now, a lot of the nutrient studies that are done, the idea is that they're looking at what's going to happen in the future with climate change as the ocean pH goes down, as nutrient levels go up. So they're trying to see predicting what's going to happen. And as that pH goes down or alkalinity goes down, what's going to happen to these corals? Sure, the nutrients go up, they're going to grow more, but they will be less dense, less able to withstand storms and things like that. So my question is, a lot of these studies have looked at, okay, give them too much nitrogen, give them too much phosphorus, but few have looked at giving them both, and very few, if any, have actually looked at what happens when you adjust the alkalinity. Because they're not looking to increase the alkalinity, they're looking to see what happens as the ocean's alkalinity starts to drop. So my question is, if I have a high NP ratio and I have high alkalinity, am I still going to get brittle corals or not? So that's something that I would like to look at in the future. So this is the graph from when we had all those corals dying off, and this was my molar ratio, and this was the alkalinity. So we had a double whammy. So we had our ratios going up, and we had our alkalinity dropping until we started to be able to get it back up again. So maybe that also had something to do with why we were losing them. So let's look at a few more slides here. Take a look at what these things do. So ammonium. There's a lot of talk in the forums, a lot of people talking about Sanjay Yoshi, Mike Paletta, other people about dosing ammonia into your tank. Is that a good thing or not? Again, as I mentioned at Waikiki, we had higher than normal levels of ammonia. So we had open systems where this well water was going into the coral tanks constantly and overflowing. And they did a study back in 1994 where they measured the level of nitrogen and phosphorus going in and the water that was leaving. The water that was leaving was 0. And these were like 4 by 4 by 2 foot containers, so packed with coral. So they were sucking it all out, no problem. So corals will use ammonium over nitrate. They prefer ammonium because to use nitrate, they've got to first enzymatically break it down. It's metabolically expensive for them to break nitrate to get the nitrogen out. Ammonium, not so much. So they prefer to use ammonium. So for me, it's not surprising that if people are dosing ammonia, it's better. However, these people usually also have lower nitrogen levels, nitrate levels, sorry. So if you have really high nitrate and you start dosing ammonia, I don't know what's going to happen. Does your nitrate keep going up? Does it go down? I don't know. So if you have slightly elevated levels, it's probably not detrimental. But if you have a persistent level, if you can always measure ammonia in your tank, it's not good. You're going to have issues, which I'll talk about in the next slide. It's much better to pulse it, so you give them a dose, and then you don't dose again. So you want to use an auto doser or anything like that to constantly be pumping it in. You give them a shot of nitrogen or a shot of phosphorus, and leave it and call it a day. The greatest benefit when dosing it, ammonium is seen if bicarbonate is also dosed, or if alkalinity is above natural seawaters or higher. So the biggest benefit you're going to get from adding ammonia as your nitrogen source is also to make sure you have good alkalinity levels. And what are some of these effects? Well, first of all, if you start dosing ammonia, you may see your corals starting to get darker because they're making more zooxanthellae. The zooxanthellae are multiplying. And it's, when I'm looking at the literature, there's a lot of variation because it depends on what corals you're talking about and what kind of nitrogen form they're using. So there's a lot of variables here. So in some cases, adding it decreased the growth rate. And other cases, it increased the growth rate. And in the latter case, when they make it increase that growth rate, the skeleton becomes less dense. Again, they never reported what the alkalinity was. They didn't do any dosing. So who knows if that will help or not. Interestingly enough, if you dose with ammonia, you increase their ability to resist bleaching. The other thing is if you have a prolonged dosage of ammonia, if you always keep a persistent level in, what they found was that reduced the fecundity, fertilization rates, and embryo development. So if you're trying to breed your corals sexually, you don't want to have any measurable ammonia in your water because it can depress their ability to produce viable gametes. And then if you're adding nitrate or if you're adding orthophosphate, PO4, the effects are basically identical for doing either one. Again, increased number of zooxanthellae. You saw that one picture where they increased it, the coral got darker. Decreased or faster growth rates again. They do see a increased rate of disease. So tissue loss, black band, white band disease. They're more susceptible to bleaching when thermally stressed or when light intensity increases, which, back just before we had that outbreak of brown jelly, just before COVID, we had just changed all the bulbs. And our whole nitrogen phosphate ratio was way out of whack at that time. So maybe that was what they stressed the corals out even more, more susceptible to that change in light intensity. Again, this is for nitrate. Ammonia, if you give ammonia, they're more resistant to bleaching. And as I said, the effects vary from species to species. So some caveats, takeaways from this. First of all, this is my system. This is my take on my system. I'm not saying this is going to apply to everybody's system, but it's another tool for you to look at. If things aren't looking right, corals are starting to get lighter in color, you're seeing more bleaching, you're seeing tissue recession, things like that. Check that. Not all genera react the same way. Turbinaria, Stylophora, soft corals are completely different. They could care less. You can add all the nitrogen and phosphorus, doesn't matter what the ratio is, they suck it up, they just grow fine. Stony corals, particularly Acropora, much more sensitive. As Christine told me when I was there in June, Acropora are just strange. They are much different, which I'm sure a lot of you can attest to. There's also the role that food can play. There's still some research going on that. In some cases you would think it would help, in other cases it doesn't seem to help. And I said there's a lot of conflicting results in the literature when it comes to the effects of nitrogen or phosphorus or when you combine them both. So what you really need to do is you need to have some context when you're looking at this kind of stuff. What's the coral tax that they were using? What was the morphology? Branching corals react differently than plating and massive corals do. What's the type of nutrient they were using? Were they using ammonium or were they using nitrate? And then rarely do they talk about alkalinity. So in our cases, we are boosting our alkalinity or we're keeping it at normal levels. Like I said, they're more interested in seeing what's going to happen in the ocean in the future. And like I said, they're based on those effects of climate change and chemistry. So increased skeletal tissue and growth increases the rate of photosynthesis and growth. So they're going to want everything else too, right? Iron, manganese, calcium, all that stuff. All these trace elements. You're going to want your corals to grow faster, they're going to be hungry for this stuff. They're going to deplete it more quickly. Also, if you have elevated nitrogen or phosphorus, you could run the risk of increasing the rate of diseases. We've seen that in our corals. If our phosphate got too high, we would start seeing brown jelly, particularly on Acropora. If our nitrates, if our ratio got out of whack, we see bleaching. And they're also more susceptible to thermal bleaching when that ratio is off and you're changing lamps or you're increasing the temperature. I haven't talked about temperature, but they're more susceptible to bleaching if the temperature goes up. And if you're trying to spawn your corals and you're raising the temperature in your tank, something to keep aware. But all these got me thinking, like all these results that they were getting, is that because they were screwing around with this? All these negative effects, was it because the ratio was too far out of whack? Because they were just adding nitrogen. That's also what Christine was saying, that a lot of these researchers, they were just putting nitrogen in. They said, oh, my corals were doing bad. She said, yeah, because they had no phosphate. They didn't have enough phosphate. All right, so enough about nutrients. I want to quickly show you something that we just started working on, and that is 3D modeling of our tank. So when you have 212,000 gallons and you have over 1,000 corals, how do you keep track of them all? How do you know which ones are growing? How do you know which ones you've got, which ones have disappeared? We gave up trying to count them probably about 10 years ago. It just became a really, because every coral had a number associated with it, and then they would get moved. And it was just, we didn't have a good way of documenting that. We would take photos and try to count them in Photoshop. It was just very, very labor intensive. So back in 2018, we were doing a video for the planetarium about coral reefs, and they wanted to recreate a 3D model of the coral reef. So they gave me this camera and said, go in there and photograph this coral from every angle you can think of. So that's what I did. So this is basically, this is a point. So every one of these points represents a surface on that coral. All these blue squares are the photos that I took. So then they would take all this information, stick it into a computer program, and recreate the model of the coral. So this is called photogrammetry. And it's also a tool that's used in research. So then they said, okay, let's do the whole tank. I said, well, I don't have time to do that. So this is Dr. Luis Roja, who went in. He's a very quick swimmer. And he took all these photos. I think he took over 400, 500 photos of the tank. And then they took all those photos, put them in a program, and recreated the tank. So this is all that echinophilia. This is in 2018. By 2020, that was all gone, all of it. And so using those photos, they created this. And then they used that to create a digital version of our coral reef for the planetarium show, which you can still see, depending what time, what day you come. So then we also have a guy, Dr. Pim Boomgerts, who's a crazy Dutchman, who's part of our research team at the Academy, who does photogrammetry in the wild. And he mainly works in the South Pacific and also in Curacao. And so this is his rig. It's tied to his scooter, and he has all these lights. He basically goes and he swims along the bottom, and he films the reef. And he dives mainly to about 200 feet, in that range between 100 and 200 feet. So he does a whole plot like this, and he can go and scan it. And so every year he goes, he can map that reef, and then this year in December they found a bunch of bleaching. So he came to me and said, you know, I'd like to try this in your coral tank. I said, well, I can't take a scooter and that giant rig in there. So he built me a rig with some floodlights and a GoPro. And a couple of weeks ago, I went in and I just swam and videotaped the whole reef, well, parts of the reef. And it was the first try, and he put it into his software program, and he created this. So now we can go in and we can count all the corals. And what's cool is that there's also some little, I think you'll see them in here, little squares that have known distances. So with the computer software, you can measure that distance, that known distance, and then draw a line on the coral, and it will tell you how big the coral is. And then as you can also put point maps. All these corals can be given points. And so every time you do it, the computer will track them and it will show you where they are. You can just push a button, show me all my Acropora millipora, and it will show where they all are in the tank, and then we can measure them. And then also once you do, if you also measure the z-axis, then you can get the volume, and you can measure the volume of it. So there he is, he's trying to, this is that little thing there, so he's measuring between the 2 points, and then he's going to go and he's going to take it up to a coral and just draw a line on it, and it will tell him how big it is. You can also do color, so you can measure the color spectrum. You can see if the color is changing and shifting. So we're just trying to figure out, okay, how can we use this and is it really worth all the effort it's going to take? Basically, I only, when I went in there to videotape, it only had like a half an hour, not even less than that. He said, really, you need to do a whole hour worth to really get all the little nooks and crannies. All these black areas are where there's no data that it didn't get. So, yeah. So that's something I'm working on now, and then also if it works out, and then we can transfer it to other institutions that also have these giant tanks like that, so they can track growth. So these are the current people on the Philippine Coral Reef team. These are all the people that over the last 16 years have worked on that tank. And photos and videos, thanks to those people for sharing that information, and for Max at Burgers and Steve at Georgia for allowing me to use their data. And then these are all the departments at the Academy that support us, that support that tank. Our dive operations, our electrical department, our electronics people, our engineering department, and our operations department. Not to mention the Institute for Biological Institute of Science, the researchers. There's some papers that are saying that the lower the level